if you just do it, it'll turn out okay. If you were with us last week, we talked about fictional spiders, starring Andrew Garfield. But today we are talking about the real deal and their cousins.
Hey, yeah, it's spider time. Let's get this show started. Talking about spiders and scorpions, and talking about some solifugids and some ticks and mites, and there's so many arachnids are so much more diverse than people think. But most people think spiders and scorpions, but there's a lot more to them than that. And in the past, there was even more. And if you want to expand this to chelicerates, you get some really weird guys like the sea spiders and the horseshoe crabs and things like that.
But before we get started, because you've been living under a rock, guest, introduce yourself.
Hi, I'm Bree from Hive Sci. I have a science and debunking channel. Mostly we focus on debunking bad science, especially when it comes to animals and biology and phylogeny and ethology and ecology and all those overlapping fields. When that goes awry, I'm here to say, hey, let's pump the brakes there, kitty cat. And I started my channel talking about bugs, definitely extended beyond there, but I still love talking about our little arthropod friends because protostoma holds a special place in my heart. I do have a collection of arachnids myself that I've hand raised, each and every one of them, from a tiny sling to an adult spider. So I know my way around. Anyway, when you asked me to do this, I told you that that I had recently put together a whole bunch of content for a video I was helping another content creator with. So I had all my notes already ready for this. So it was fortuitous that you wanted me to come on and hang out.
Timing. It's always good to have good timing.
Mm-hmm. Yeah, and Ness, I believe, might show up at sometime during the stream. I don't know if they're still if they still plan to. If not, we'll just keep rolling. All right. So, what is an arachnid? That's a good question, huh?
Well, this is what I found out. Apparently, this might be the wrong list, but apparently there are at least 5 characteristics of arachnids.
Yeah, this is a good basics. Sure. Let's see. We got body segmented, usually consisting of a head, thorax, and abdomen. Yes, a lot of arachnids have a fused head and thorax called a cephalothorax. That's what you see in spiders. And opilions, those are harvestmen, the daddy long legs that look like spiders. They actually have a fused head, thorax, and abdomen. So they just look like little buttons. They don't have jointed body segments like their ancestors did. Now, other groups, of course, you have like the scorpion, which has its abdomen elongated into a nice sturdy telson to help it with its feeding process. Let's see next. We got jointed appendages. So this is definitely something so far that I can agree with. It's all characteristics of arthropods. Okay, I'm with them there. And firm but flexible exoskeleton are also a good feature for arthropods in general. An open circulatory system. Yes. Specialized appendage, including claws, crustaceans, or wings. So that's a weird thing to say that about arachnids, because there's no arachnids with wings. There's no.
Yeah, I was wondering why the insects. I thought insects were different clade altogether.
Yeah. So insects and crustaceans, they're the other major clade of arthropods. They're in a group called Mandibulata. So that they're actually it's split in two. And if you want to get technical about insects and crustaceans, insects are crustaceans. They're just hyper land developed crustaceans. But
But if you want to go in a deep dive about our talk about a certain clade of insects, go check out mine and Bree's episode on whatever the wasp name is.
Hymenoptera.
Yes.
So I was saying it's divided into, there's two major groups. There's the Chelicerata, the Mandibulata. That are the two major clades of extant arthropods. There used to be a lot more Radiodonta and all kinds of stuff, but right now we mainly have either Chelicerata or Mandibulata. Nats, you're cool. It's okay. People forget stuff. We're cool. Just join if you want to, and if you can. Love you and thank you for offering to pop in and entertain us with your big old brain. Anyway, so crustaceans and insects or Mandibulata, they're not going to be on the menu today. So we're looking at Chelicerata. Now, Chelicerata, you got to narrow that down more too, because we talked about there's sea spiders and the Eurypterids, the giant sea scorpions of the past. And then there's horseshoe crabs, all different clades of Chelicerata that are not arachnids. Arachnids are evolved for terrestrial lifestyle. So there's no primary evolved aquatic arachnids. They're all, if any arachnid has aquatic characteristics like a diving bell spider, there's secondary adaptations to help them make that transition. But arthropods did develop wings in the insect clade, but there's never been a Chelicerate with wings. Now, that doesn't mean they can't fly. Baby spiders can fly. They use a silken line that they navigate with the Earth's magnetic field. Pretty cool stuff. And they're not just floating around. They're navigating. It's pretty nifty. Welcome, Nas. Good to see you tonight. I guess it's.
Now you can make it as long as you can make it.
Yeah. All right. Yeah, here we go. We got a breakdown of the cladistics of arthropods. As you see, the first branch off to the left leads to velvet worms. Now, velvet worms are they're very closely related as a sister clade to arthropods. They're also a sister clade. If there was another branch off of here, it would be for water bears or tardigrades. They fit right next to the velvet worms. But when you get into that middle branch right there where it splits in half, that middle split is the split between Chelicerata and Mandibulata. Mandibulata is on the right. You got myriapods listed in there as well, which are centipedes and millipedes. And also, I believe remipedes fall into this group as well, but they're more distantly related to the rest of the mandibulates. Then we get into the clade that we're talking about today. You got Chelicera in the center left here. And as I mentioned already, sea spiders and horseshoe crabs, not arachnids, but that brings us down to this final little branch right in the middle where it says arachnida with scorpions, spiders, and more. I cannot hear you, Ness, if you're talking.
Nope.
No.
We cannot hear you. So sea spiders aren't true spiders?
No. Sea spiders are just their own thing. They're really weird too. They're like minimum body proportions. They're wild. They're like 95% of their anatomy is legs. Even their lungs are in their legs. They're so unique.
Got the name, but not the clade.
Yeah. Well, and horseshoe crabs are not crabs. They're not even mandibulates, which is where crustaceans are. So they get misnomered too. But we got sea scorpions as well, which aren't scorpions, and they're also chelicerates.
Oh yeah, they're starts with an E, I think.
Eurypterids.
Yes.
Yes. I think we had a slide of Eurypterids if people want to see what they look like. Did you have that one or not? I'm not
I,
sure which ones you were able to utilize. I know there's a lot there, and some of it's more technical than you wanted to go because we're doing a broad overview, not with R, and it was like a 4-hour thing, so there was a lot to go over. We're not going to go 4 hours.
uh,
uh,
this is the first part of your notes I copied over.
okay. Here we go. So we talk about arthropoda. That's the source where arthropods come from. The Cambrian, the third stage, average temperature is about 70 degrees Fahrenheit. That's 21 degrees Celsius. I wouldn't use this slide though because this is just giving instructions about how to address the topic.
Okay.
Yeah,
Right.
I was providing this to you for these pages to you for the images, not necessarily all the notes.
Okay, then
yeah.
Hold on, then let me get that.
That's cool. Yeah, the Burgess Shale's full of lots of early arthropods.
Here's
though.
your first.
There we go.
Here's your first picture.
Okay, so again, we see the divide between insects and chelicerates here, giving us a broader spectrum of what falls under the spider clade here, but because they always started off with spiders when we're talking about arachnids, right? It's always spiders, then scorpions, then mites. Even though spiders are relatively newcomers on the scene when it comes to arachnids, whereas scorpions have been around since they were still living under the sea. Yeah, but they've been replaced in their niche under the sea by crustaceans. So that's why we don't really have any sea scorpions left today. Any aquatic or marine scorpions, because scorpions used to be actual arachnids that lived semi-aquatic lifestyles. True scorpions like Pulmonoscorpion kirkinensis is the first scorpion that we're sure lived on dry land. Everything before that was either aquatic or semi-aquatic.
Am I audible now?
Yeah, we can hear you now. Woo!
Finally. Woo. All right.
Yeah,
so
this one
my apologies for coming late. Sorry about that.
You're all right. I'm glad you're able to make it. And this one has anomalocarids on here too. It doesn't have so we're talking like radiodontia being off that branch as well. So yep.
Yeah, the radiodonts were the first predator, like apex predators of the Cambrian.
Yep, early conquering arthropods. Really, that was the clade that put arthropods on top. Right before then, it was just little tiny things. Hard-bodied organisms were pretty basic trilobite-like. Everything was from what's the one I'm thinking of? It's like a trilobite, but it's feathery. Eek. Morelia. Yeah, Morelia. That's what I'm thinking of. Yes. So everything had that simple body form. I think trilobites are sea arthropods, if I remember correctly. That is true. They are another clade of arthropods. That was dominant back during the Paleozoic era.
And sadly, they get past the Permian.
Few things did, though. So, I mean, we did. Go us, right, guys? Woo!
Yep. We made it past all the big five.
Yeah,
I think it's also a debate. There's also a debate whether trilobites are closer to the chelicerates or the mandibulates.
Yeah, I know. I love that topic too.
Yeah.
That would be. I'm more in camp mandibulata, and I'll tell you why some other time because this is about chelicerata. But
yeah,
I think that trilobites.
You gotta pay for that episode.
Are the Eurypterids closer to the horseshoe crabs or closer to the arachnids? Do you know that?
Not right off the top of my head. I don't know. I wouldn't want to make that assertion. There's a lot when it comes to internal clades within Chelicerata and how they're related to each other. It's so disputed, and when things evolved, and what counts as a spider, and what counts as a scorpion, what counts as an arachnid, even is so so hotly disputed that it's hard to pin it down. Anyway, what's our next slide here?
At least according to Wikipedia, Euripteryps appears to be the sister clade to arachnids, so they are more closely, at least according to what Wikipedia shows. But still, yeah,
that's the current findings. Then, yeah, I'll go with that until something proves otherwise. I'll accept that. Yep. All right. Let's see. So now we're talking about just the different appendages that these creatures have. So chelicerates, they emerged from arthropods in the Middle Cambrian about 500 million years ago, and like all the other arthropods, it was in the warm, shallow seas that they were really able to thrive. Right, some of them actually started leaving the seas really early on, about the same time, like in the early stages of the Paleozoic. Now, apart from their distinct chelicera, they also came to be distinguished by their unique brain structures because they lack a frontal portion of their ganglia that converged in to form the brain in other arthropods and in our ancestors too. They never really did that with their evolution never really took them down that route, so they never developed what a brain really is. It's more, they have more of a system of extended ganglia, and it's segmented down their body. You see how these images have it segmented here. These traits, they go hand in hand with like development in other arthropods of like antenna and stuff. That's why you don't see arachnids with antenna, because they don't have that sensory part of the brain that was used to make antenna useful. Like we see in like insects, isopods, centipedes, so all the mandibulates that just never rose in chelicerates. We'll see a myriad of ways the chelicerates compensate for the lack of this sensory part of the brain. And I think one of the main ones that everybody can remember and kind of relate to is the whip scorpions that have the long front legs that are just basically turned into antenna that feel around in the dark. So that's a tactile alternative to having a chemosensitive antenna like a centipede or an isopod or an insect has. Now, there are some genes that we could talk about here when we're looking at this image right here, because this is going to be relevant. Oh, go ahead. I'm sorry.
Sorry. The first picture up there, is that only picture? Is that like the apex predator of the Cambrian?
So these images aren't specifically any species. It's showing the development of how these Hox genes. See the feathery appendages on some of these images?
Yeah,
those are lungs, or their breathing apparatuses. So the Hox genes in these organisms developed to help them breathe through these. Each segment of their body has intake for its own oxygen, which gave them a advantage over other organisms that were competing in their environment, because they were able to maintain a more active lifestyle. Because they could, as they move through water, they could siphon the oxygen from it, unlike like trilobites and stuff.
Just the arms remind me of that. Mm-hmm. I can't remember its name now.
So we're looking at when it comes to the gene development here, we're looking at do you all want to know names of genes and stuff, or you just want to
sure.
I mean, we're looking at SP6 through 9 and D2. Those are the embryonic genes to develop legs.
And you want to know that? You might want to know more. You can go. You can look that name up now.
Mm-hmm. And the genes that were used to develop legs. Remember how we talked a minute ago about how they didn't have that ganglia in the front of their brain? So what they've done is they've used those genes for legs and put those in the front instead, where every other animal has mouth parts and maybe antenna. They have leg genes, which create things called pedipalps, right? That are pseudo legs. They're not really legs, but they're kind of like legs. And their chelicera also pulls in these genes to develop the chelicera, which are also like the leg genes, but in spiders they're fangs, and in different groups they have different functions. But really
And then
interesting. Go ahead.
like in basal groups, they are more like claws, but they pinch.
Mm-hmm. Yep, because scorpions are the most basal. Yeah, they were claws. They were claws first, right, on all of them.
Yes.
Yep. Those genes have kind of pulled back to be replaced with these Hox genes for leg development. Now, some early arthropods had crazy leg development. We talked about radiodontids earlier. They had Hox genes that just went nuts. So they had like 14 pairs of legs and things like that. But with arachnids, it was whittled down to 8. But then they decided, again with this, and I say decided in a colloquial sense, but it was they developed these facial legs to supplement not having this front part of the brain that all the rest of the arthropods had. I always thought that was interesting. There's their distal leg genes is what they call them. But, now let's move on to the. Go ahead.
If I try and flip super fast, in your personal list, what got you interested in arachnids?
So I always was like collecting and playing with spiders when I was a kid. I've always had spiders as pets. I was really into animals in general, and I wanted to have my own zoo, but that's not viable or good for the animals. But spiders and arthropods really they don't care as long as they can thrive in an environment that mimics their wild outdoor counterpart. They don't care about being in captivity. So I have my own little zoo of spiders and insects and arthropods. So yeah, that's what I did. I made my own little zoo. All right. All right, we'll be looking here. Okay, so now we're on some fossils. So here we're looking at some more of the early arthropods as they or chelicerates as they were developing early in the aquatic and marine environments. This is the era when they started to move on to land. Unfortunately, I don't have my notes on this particular slide, so I apologize for that. I don't know which species we're looking at here. I did have.
Let me check.
Go ahead.
Well, hon, I'm still copying the pictures over, but 1 second.
Yeah, sorry, I wasn't clear about this just being, because this was literally just like notes on how to address the topics and stuff. So,
well, there's your document you had. I put in private chat. You check it out.
Yeah, well, I got it open here on my other screen. I already got it pulled up, so I got the notes, but it doesn't have the some reason when I copied this over, I didn't get the subtext for this particular image. I don't know why I forgot to add it, but I don't want to misspeak about what we're looking at here and what species these are. But I do know that these are a series of early chelicerates known from the fossil record throughout the Paleozoic era, that when hard-bodied organisms started to get a lot bigger and leave more robust fossils later in the period.
So what is the oldest? What the oldest fossil of arachnids are?
Well, as far as what is an arachnid is what we're going to get into there. It's really it comes down to where you want to draw that line, because there really is a robust transitional lineage that we can watch go from generic Chelicera or Chelicerata to specifically arachnids. Every stage along the way is accounted for. Now, let me see here. We're getting into
I
way
think
too far ahead of myself.
I think there are also many groups too that have a very poor fossil record. Like we don't have a lot of ticks, I would assume, right?
Yeah, because they've always been small. We do have ticks from like dinosaurs and things like that, though, which is really cool. Like a tick that was on a dinosaur feather. I actually did a response to somebody saying, "Oh yeah, we don't know for sure that it was a dinosaur tick." I'm like, "It was found on the dinosaur feather. What are you fucking talking about?" Sorry, language. I don't want to get your channel demonetized.
Don't worry, I'm not monetized to begin with, so feel free to cuss away.
Oh, I thought you were monetized. You have. Me and you have about the same amount of subscribers.
Yeah, it's half subscribers, but half view out. You also need the view hours to counteract that.
Oh. Okay, I get you. Where are my notes on ancestor of all arachnids? Here we go. It is estimated that 98,000 arachnid species have been. Okay, that's the OG arachnid body plans. The scorpion. The oldest scorpion is Dolichophallus. It is 436 to 428 million years old is how old the fossils are. So that answers your questions as far as being a true arachnid. Somewhere, go ahead.
So I said, all right.
Yeah. And then there are some genes that, let's see, I already talked about that. About the leg genes.
All right, 1 second. I need to reset this slide real fast. So give me a second.
Yeah. Well,
what's the oldest scorpion and oldest spider that we have in the fossil record? Do you know that?
The oldest, and that's another kind of questionable pedigree there. What makes it? What really makes a spider? Because we have things that are for all other purposes. Everything about them is a spider, but they don't have the silk spinning genes. Now there are other schools of thought that would put like tarantulas, like my gallimorphic spiders, outside of spiders, so make them not true spiders. But we have things that are sort of a halfway point when it comes to spiders specifically. We have things like Chimera arachne, where it has a telson still, but it's definitely more spider-like, and in that spider lineage.
It did, like Chimera arachne did produce silk, according to the fossil, but it has a very like ancestral traits for arachnids. Like arachnids started out, as you said, they originally had a telson, like a long quote unquote tail on the back end. It's not a true tail, but you, like layman would say, oh yeah, that's a tail on the back of the animal.
It's made up of metasoma. Yeah.
Yes.
Which is what you see here is also the same condition in these early chelicerids that were maybe arachnid. These are the guys. These are the guys that are like, hey, are these arachnids or not? You can see that they're very horseshoe crab-like. They kind of toe that line. It's funny because people expect earlier arachnids to look more like eurypterids. but
You
they
mentioned
came in all shapes and sizes. Go
They
ahead.
also look very much like trilobite as well,
I know,
almost.
I know that somebody didn't one of the creationists. That's one of the creationists saying that horseshoe crabs are really trilobites, lasted forever, and they're still alive. See,
Yeah, well, they'll look at like isopods and make that claim. They'll be like, look, isopods are trilobites. They're the same thing. It's like, no, not quite. But these guys definitely give rise to the Chelicera line, right? We're looking at the Ordovician. These are the early pioneers, some of the first organisms that are thought to have moved onto land, even and using early marshes for spawning and things like that.
Mhm.
So pretty interesting ecology that they find these critters in, with lots of semi-marine stuff and semi-terrestrial stuff. So I really am watching.
Glad. These are glass pits, right? So are they their own group, or are they like a paraphyletic group?
They're primitive chelicerates is the best way to describe them.
All right.
They
So all living chelicerates are more closely related to each other than they are to these glass pits.
Yes, yeah. Even like between horseshoe crabs and arachnids, they're close related to each other than these guys. Even though there's the similar look, and you expect the original body plan to be something more like a Eurypterid, but it's really more like a trilobite.
So are these guys more or further closely related to each other than we are to tuna fish?
What?
I think Vandal is asking, like, are we more closely related to tuna than the spiders are to the glass pits? I think it's almost the same. Maybe I don't know exactly when they diverged.
So
there are some. It says right here there are some Cambrian types.
Yeah.
So they diverged earlier. So I think we are closer to tuna than spiders are to these guys. I think.
I think all tetrapods would be close related to each other than these guys would be to any modern chelicerid.
Yeah.
But
that shows the diversity these guys have then.
These are the types of organisms, though, that they will throw up on the screen and say, "Look, here's a horseshoe crab that hasn't changed," and they'll show you. They'll show you these, and it's like, these are not horseshoe crabs. They're not the same. But not to say horseshoe crabs aren't ancient. They are, but even the horseshoe crabs that are ancient aren't unchanged, right? There's adaptations that they have, but they're just really good at their niche. They're really well fit for their niche. That's all that matters. You can hold on to it for hundreds of millions of years if you're really good at it.
This is my territory.
Shark.
This is my territory, and you can't have it.
Mm-hmm.
Get off my lawn.
Also, it's like, as this image shows, that the Cambrian glass pits have eyes, but in the Ordovician they lost eyes. Like, were the later ones like deep sea dwelling and they lose their eyes, perhaps? I don't know.
That's an interesting speculation, and you wonder if maybe they lost their niche in the more shallow waters to something else, to Eurypterids and more derived. I think there are radiodonts still around at this time, right?
Well, very few. There's like one famous filter feeding radiodont that survived in the Ordovician. Like, that's one of the latest survivors. I think most of the predatory went extinct in the Cambrian, I believe. Yeah.
Yeah, I think I know the one you're talking about. The Cories, Mecha Cories or something like that. Is football sized, but pretty big for the time that it lived in. No. All right. So this is just giving us our basic body plan of an arthropod, of a chelicera, and what sections of the organism that we were looking at, including the telson that we were talking about earlier. This one's not divided into metasoma, though. Metasoma is when the telson segments itself into different. They'll just be numbered: metasoma 1, metasoma 2. This one's just got the 1, but some of these body segments in early chelicera were there was like a whole lot of segments, like what you see in more basal arthropods in general, right? But as they derived into different niches, they started to fuse these body segments together. What's interesting about that is those leg genes that we were discussing earlier. They would fuse and double up the genes like that too. So one leg is a clone of the leg on the opposite side, like exact clone. And if it loses that leg, it doesn't replace that leg with genes from the leg that was lost because it's gone. It replaces it with genes from the other leg, so they can actually manipulate. PZ was talking about this too. They can actually manipulate the genes in one of the legs to make it develop weird. I thought that was cool anyway, like make it fork at different joints and things like that.
So
I have a question about the telson. Like in scorpions, is the entire length of the like the stinger the telson, or is it more than the telson? Is the telson in the scorpions just the tip of the stinger?
So the tip of the stinger is what they call the telson in the anatomy of a scorpion, but the whole metasoma, all the metasomal segments are formed from the same genes as the telson.
So it's like a lot of the tail, quote unquote, of the scorpion is also the telson, basically.
Yeah. Every segment, they're called metasoma or metasomal segments, and then they're numbered. Scorpions have 5, I think. It's because it's 5, but 6 if you count the telson itself. So
now I know you're going to get to it later, but do these guys make a big impact in the Carboniferous like the insects do? Are they grow big sizes too, or?
Yeah. Oh yeah. We got Pulmonoscorpius. The biggest scorpions that ever lived during the Carboniferous. Some of my favorites, and they were big. They weren't as big as the ones that lived in the water.
Yeah.
Eras, but as far as land dwelling scorpions, they were the size of. People say they were the size of a cat. I would say it's better to say they were the size of a squirrel.
That's still big for them.
Yeah, because if you think about the way the tail curls and everything, yeah, the weight, they'd be lighter weight, and
I am glad. However, I don't know if you're glad or not, but I guess I'm glad that we never reached the old seaside, the big size of the scorpions underwater in the past that do on land in present day.
Yeah, be like Clash of the Titans, fighting giant scorpions on your way to school every day. Yeah, so the segments here, what's calling post abdomen, segments 5 through 13. Those are the segments that are going to fuse into the metasomal segments in the scorpion. Now, all modern arachnids came with that fusion already pre-installed. They don't have this many segments. They really only have the abdomen, and here it's called pre-abdomen. They have this cephalothorax in a lot of arachnids, but that's gonna be a recurring feature that we see in all arthropods that we call carcinization. That means the more specialized they get into a niche, the more likely it is for future generations to lose body segments along the way. The more and more specialized they get to a niche, the more segments they lose, which is really fun when we get down to, like I was talking about at the very beginning, harvestmen. which only have one body segment left. They're just the butt and the legs.
And there's also a very misconception, like harvestmen are not spiders.
And they're not venomous, right?
Yes,
No
also, yeah.
venom. And they're one of the few arachnids that can be that'll eat
I think
plant
the group is
matters.
called Opiliones, right?
Yep,
I think it's true.
Opiliones.
Yes.
Yeah, we
And
talked about.
like they are closer to scorpions, I believe, right? Or are they than the
Yeah.
spider? Yeah.
Yeah, so they are and they're a lot more diverse than what you see, in your backyard. Till you get down to like jungles and stuff. Oh my gosh, they're so beautiful and diverse. Some of them are ornamental or ornamented. Some of them are colorful. Some of them glow in the dark. I used to have some glow in the dark ones when I was younger, but I should get some more of those.
Just like
They're pretty cool.
scorpions. Yeah, scorpions also glow in the dark for some reason. Yeah.
Yeah, there's something that helped to do. I imagine it's something to do with their nocturnal behavior and their need to absorb certain wavelengths of light for their metabolism is my speculation, but I don't know for sure. It's all speculation on my end.
Yeah,
It's
before you
still a
got.
big mystery. Yeah, it's still a big mystery. Maybe there is no maybe there is no reason. There's also a possibility that maybe they just acquired like a fluorescent protein and it's just stuck around for some.
Yeah,
Also
like did you
possibility.
see that they put the glow in the dark gene in the cats?
Yes.
It was just a
yeah.
byproduct. Yeah.
Yeah, before you got here, we were talking about how sea spiders aren't true spiders. Now names can be. They give them names, but they're not. The people name things, and later on, look, these aren't related at all.
It's folk taxonomy gets a lot of things off when it comes to arthropods, especially. Like there's little less confusion when it comes to like vertebrates. Like a fish is a fish, although even though that that has problems too, ironically enough. But it's like it's things that aren't spiders are called spiders. Things that aren't scorpions are called scorpions, and
things that aren't lizards are called lizards.
Yeah. Also, yeah.
Well,
unfortunately, it's first come, first serve names.
So there's the major ones that you think of when you think of arachnids, and those are mostly the tetrapulmonata, which is scorpions, spiders, amblypygids, and thelyphonids, which is like the whip scorpions. Then the amblypygids are the tailless whip scorpions or cave spiders, or whatever you want to call them. Those are the tetrapulmonata, and then outside of that, you have the pseudoscorpions, you have ticks and mites and acaria, and then you have closely related to mites actually solophugids, the camel spiders. And you see that with how furry they are, how fuzzy they are, and how soft their bodies are. They have a lot in common with mites when you really look at them.
Oh yeah, like it's very it's if you blow up a mite, then you can see the resemblance between a fulgid, yeah.
So Bree, if you go back and rename something, what would you rename something? If you go back in time and rename something in the tectonic record or cladogram, what would you rename?
Oh my gosh! If it doesn't have to be an arachnid?
No, like anything you like. That's a bad name for that thing. Like what they do.
No, I would go back and change Basilosaurus to Zeuglodon if I had an option to do anything like that. I hope everybody would. So I think calling it a Basilosaurus is
according to I see a phylogeny on Wikipedia for mites, and it says that the mites and the ticks are separate groups. They are not monophyletic.
Yeah, you got Parasitoforma, and then there is another group that's got the Racinonuli and Solophugids and Opilions, and you got Acariforms, which Acariforms are also mites. They're the little red guys. And then pseudoscorpions are their own thing too. Yes.
They are closely to the Parasitoformes, the ticks.
Mm-hmm.
Yes, the false scorpions are closely to the ticks, and you have the sulfurids are closely related to the vegan mites that don't suck blood, or at least some of them actually do suck blood too.
Yeah, well,
Mites,
some of them are
yeah.
detritivores and some of them
Yeah.
are predators. So there's a
We
few
have a
different.
question for you.
Yeah, so scorpions' claws are not venomous. There's no venom in any claws of any arachnid that I can think of off the top of my head. I don't think any of them would have what you'd call venomous claws. No.
The claws in scorpions, are they the chelicerates? Or I think they are not the chelicerates, right? Or are they?
They are
claws in
they're
scorpions.
the developed pedipalps.
Yeah, in scorpions, I think the big claws are the pedipalps, right?
Yeah, which is not convergent, which is analogous with what developed into the fangs for spiders. Now,
No, I think in the fangs in spiders are the chelicerates, or the chelicerates.
oh yeah, I got that. Sorry,
Yeah.
I'm mixing
Yes.
myself up here. So
Yes.
they're in spiders. They developed into the reproductive.
Yes.
With
Yes.
with the bulbs on the end. Sorry, it's been a long day.
Yeah, the pincers in scorpions are the pedipalps. Yeah.
And they have but their chelicera are also little pinchers if you look.
Yeah, yes, they are. And they're very, very close to the mouth. That they chew the food before they digest, they ingest it. Yeah.
Very much analogous with the mandibles of Mandibulata. Ironically enough.
Speaking of claws, here's some pictures of.
Yeah, look at the size of these Eurypterids. You got Jackalopterus on the far end in the blue.
Real fast. You said that Eurypterids aren't true scorpions, but are they arachnids?
No, they're not arachnids. They're
okay.
A sister clade to arachnids.
Yes.
They're closely related though to arachnids. They have a lot of things in common. As you can see though from these models, they have specialized back legs that are paddles for swimming. You can see a lot of convergence with what would come later from the tetrapods that return back to the sea and how their body plans would develop with the large paddles on either side to help steer and navigate in a large flat tail. These guys are basically built like manatees if you look at them. That's what's so cool about them to me.
Most of them have these like these fluke on the end, except for the pink one, the one on the second right, the pink one. It has a more of a stout shape.
But unlike manatees, I doubt any of these ate plants.
No, they probably were eating small crustaceans and stuff. They'd grab them with those claws and crack their shells, and
those claws are the chelicerates, right? Like the claws in
yes, in Eurypterids, these are the chelicerates. They're the same structurally as the chelicerate in scorpions and true scorpions.
Yeah,
but in true scorpions, they're just so tiny. They're just these same type of claws, but just super tiny. Eurypterids also have
different appendage.
These guys might have hunted our ancestors.
Probably, I'm sure they probably did. Anything soft, that little sucky mouths, like a sucky hole. And that's the main thing that I like about these guys is that they filled this niche where they have the same body plan as things that would come much later. I said tetrapods, but there are other organisms that filled this niche too that aren't tetrapods, I guess. Not many, but some.
Like would these guys swim to catch their prey? Or I think some of them may have, because otherwise, why would you have a fluke on the end?
I don't think any of them were particularly fast. But I imagine if it hadn't been for that big catastrophe, that we might have seen. Because you can see the tail starting to fork off on the Jackalopteryx on the end there. See how it's starting to lobe out into two separate paddles. That's when you start moving a lot faster, right?
Yeah, I might be wrong in my history, but I don't. When these guys were in their heyday, I don't think our answers even had jaws yet.
No, we had like a notochord. We didn't even have bones. Well, did we? I don't know. Hold on. I might be misspeaking.
Oh, at that time, vertebrates had bone,
Mm.
but only on the outside, like they had an armor of bone, and the inside
Oh,
was still
so
still
the
cartilage. Yeah,
Rhaptorigians then. Is that what we're looking at?
and we were like they were armored, but still jawless fish.
Okay.
I think during the Silurian at least. I think at the end of the Silurian, then you have the first appearance of like jawed
Then we could
vertebrates.
fight back or bite back.
Yeah, I'm looking at the Silurian right now, trying to see what kind of final exchange they had with from land to sea.
Of
See if
course.
there's any land. Sorry, go ahead.
So according to this chart, even if we were alive back then, we would have a problem because all these things were bigger than any of us as fully grown humans.
Oh yeah. Well, I don't think that they would have been. This is the thing that I talked to when I was on helping someone do a tier list of prehistoric animals, and it got to who would win a Neanderthal or not Neanderthal, an Australopithecine versus an Arthropleura. I don't. Unfortunately, despite the hype you might see in fiction and like even these pseudo documentaries, these arthropods are not like incredibly like strong. Like you could probably overpower one physically.
Yeah, it would have won the one probably. But I don't know if
Mm-hmm.
they got you if they got you a sneak attack.
Yeah, you could get swarmed by something, sure, but yeah, it's like their claws because their claws aren't evolved to deal with large prey. They're the biggest thing in the water. Those claws don't have to be strong and stronger. Well, I mean
Yeah,
the claws on most.
I say back when we were little filter feeders, then yeah, we're no match for them. But maybe now we could pick up a fight.
Yeah, the claws on most of these guys were thought to have been crushing, like hard-bodied, like crustaceans, and they're not crustaceans, but trilobites and stuff, things filling that niche. But yeah, they probably would not have passed up on snacking on your ancestors if given the opportunity.
So do you know which of these which each of these are, or each of these species are?
Well, I mean, they're all different species of large aquatic eurypterids. Jackalopteryx is actually the biggest. I've seen the fossil list in the Field Museum, but yeah, just slightly different niches and different time periods. No, I don't have these like laid out to which one lived when and what type of environment they lived in or anything. Just laid out by size for us here.
We just know that none of them are around anymore.
Yeah. This is not chronological. This is specifically, I believe this is mass based. So, yeah. Yeah, talking about some of the body parts on these early chelicera. Got.
Wait, like I think you also have seen this study like a few years ago. There was a study which says that the horseshoe crabs are within the arachnids.
Yeah,
Yeah,
they do. They go in and out, and different people will do different different.
It goes back and forth, and for now, I will say that they are not inside. It's the safest assumption for now.
Yeah.
I'm still open to the possibility, but it's still uncertain.
If they are arachnids, they are the most basal of arachnids. I'll say that they still have a lot of conditions that that all the other arachnids left behind in the Paleozoic, like all them damn legs, because other arachnids don't have quite as many legs as a horseshoe crab.
It's pretty weird because the like in that study where they uh,
Mm-hmm.
placed the horseshoe crabs within the arachnids, they
Mm-hmm.
placed it pretty deeply in the arachnid group. Like the closest relatives are the Rhizae nuclei and also the solvigids. So according to them, they are very close to the
Whoa.
horseshoe crabs are close to. Of course, I'm skeptical
Xiphosaura?
about that. Yeah. Xiphosaura, yes, the horseshoe crabs.
And they placed it like as a sister clade, like within the like closest related to again these the solifugis as we covered in a
Yeah,
previous slide.
according to this study, the sister group is called the Rhizae nuclei. It's a
Yeah, which
very
is
obscure group, and the next closest related group is the solvigids and the Palpicredi, another
yeah.
obscure.
So yeah,
Yeah.
when we were talking about the solifuges being closest related to mites, that's the group, the Reclinui or whatever.
Mm-hmm.
Right? I think that's what it was.
The Raikai Nui. I don't know how you pronounce it correctly, but it's a very they
Yeah.
are the common name is hooded thick spiders. That's the common name. Hooded thick spiders.
Yeah. And they're velvety. Like a
I
like
don't
a
think they are. I
solifusion.
don't think they are mites. I don't think they are mites. They are not
No,
mites. No.
they're closer related to
Solvages, yeah.
solifuges. Yeah, yeah.
Yeah.
There's also little tiny arachnids that are closely related to whip-tailed scorpions that are so tiny that are in their own unique clades and look like pseudo scorpions but are their own thing. Yeah, hit that like, share, and all that good stuff. The claws remind me of scissors a little. Yeah, some of the claws are designed for shearing, and some of them for crushing. Some of them for holding on to things. So it just depends on what they're doing. A good rule of thumb with like a scorpion, though, is that if it has a fat tail and small hands, it's very venomous. Uh, if it has big hands and a skinny tail, it's probably going to be all right. If it stings you, you probably won't need to go to the hospital. But you know, in my opinion, you should always go to the hospital because you don't know what kind of allergic reaction you're going to have. But that's just me.
Mm-hmm.
Have you ever been stung by one of these a scorpion or a spider before?
I've never been tagged by an arachnid, only insects.
Yeah, I've been stung by a yellow jacket once.
Mm-hmm.
When I was like about 8 years old, I think, like a very very long time ago.
I put on a pair of pants once that had a wasp in it, and it was an experience. I'll just say that. The wasp wanted out, and I wanted the wasp out, and I think the wasp thought I wanted him in there, so he kept telling me to let him out by showing me how much he hated being in there, and it was painful. Couldn't get those pants off fast enough, but this was back in the early 90s when my mama had me wearing tight jeans and a freaking belt and stuff.
Did you both survive?
No, I think he actually did not survive.
Well, I'm pretty sure. I think you survived at least. I think.
Yeah, I believe I did, but I could be that we transferred souls. I could be the wasp, and Bree could have died that day.
And that's how your love of insects happened. You became one.
Yep, that venom soaked into my pores.
I do see that like again, as I mentioned before, the study that put the hooded thick spiders as sister to the horseshoe crabs, but there was a response study that put the hooded thick spiders alongside the harvestmen. So yeah, it's a lot of discussions around exactly where groups belong.
Yeah, this is something we did run into when I was working with Aaron on this stuff too. I was like, yeah, a lot of the stuff's up in the air. What is a spider? What is an arachnid? That's all not even clearly defined at this point. So,
I think they're also very, very understudied. Surprisingly, even though spiders are very cool, even within spiders, they are very understudied.
So did you help Aaron with his this section of his phylogeny tree?
Yeah, yep, I sure did. The whole arachnid branch, and if he decides to do a insect one, we'll probably help him with that too. I'm just really excited to be a part of it. Now, I want you all again to I keep harkening back to this, but I want you all again to look at this body plan and then think of like an aerial shot of a whale, right?
Mm-hmm.
It's
They have
amazing
paddles. They have almost like flippers. Yeah.
Mm-hmm.
Did they use these paddles for power or mostly for steering? These do you know, Brie?
I think that the depending on the species, but the paddles were the paddles. I guess calling them paddles is fine. They were mobile, yeah. And so they used those for powers and the tail for steering. But I think that some of the later species that might have flip flopped, but I'd have to look into that to be sure.
I think it would be more efficient to use your tail for power and then the front for mostly for steering. Yeah,
and a more derived, a more derived one like this for sure. I think that in the way that this body plan corresponds with a whale, I can see it moving its tail up and down as a way of locomotion.
Now, have either of you seen a series on, I forget what it was on, but it was called Animal Armageddon?
Yeah.
Yeah. I remember when these guys were fighting those giant Nordicord things, cones for dominance in that series.
Oh. Sorry, I'll be right back. Oh.
All right.
So,
I would
I
think
just wanted to close the door.
I would think there are some relationships that are firmly established. Like I think it is fairly certain that spiders are more closely related to the let's see the Ambly PGI than they are to scorpions, right?
So
that's pretty short, pretty certain. Yeah.
Oh, hold on, I lost my place here. So where was it?
I can also see here that you have the tailed or the whip scorpions. The whip scorpions are more closely related to spiders than they are to the scorpions. Yeah. Like those things are certain, but they're like a half dozen obscure, like very obscure arachnid groups that are very understudied. Yeah.
Yeah, there's groups that are close related to spiders that are not around anymore, but they're different. Like how do you say this? Trigonoturbidia.
Yeah, I see it here. Yeah.
You got and then you got things like Haptopoda.
I think it's pronounced Trigonotarbids. Yeah.
Trigonotarbids. I'm not mocking your accent. I'm trying to say it right.
Like at least that's how I would pronounce it, but I don't know the correct pronunciation.
Ooh, are we getting into the embryology now? This is some weird and fun stuff. You can actually see the embryo developing its legs and its breathing parts. This is really cool. So the upper left is the early. The upper left is the early stage of a protostome embryological development. The lower left is the development of segment growth. The right shows the segments as they develop. The primary nerve system that controls the ganglia is shown in red. The blue is the digestive tract. Each segment is developed from precursor cells, also called a blast cell. I think you guys know what a blast cell is, right? Or simply a blast. They are a particularly differentiated cell, usually referred to as an unipotent cell that has lost most of its stem cell properties. Each segment, as it develops, mirrors the previous one. This will include Hox genes mirroring the set before them until they start to specialize. And then they I think the next slide should show how some of this stuff specializes. But if you got questions about what we're looking at here, uh, the coolest thing about this is when we watch the embryological development of any arachnid, any arthropod, really at the early stages. The coolest part about it is right after the one on the image on the right. Right after that, you start to see the fusion happen, but early on they all look like little centipedes. They all have a full length of little leg nubs or proto legs that are going to fuse. It's really cool.
Yeah, and a lot of embryological photos they mainly show vertebrates, but we don't show have a lot of embryo footage comparisons for this side of the family tree.
Right. Yeah, you don't get to see a lot of protostome development because we are deuterostomes, so everybody wants to talk about deuterostomes, but protostomes are really cool in the way that they develop and the way that they segment, and this is why they can do certain things that we can't, like grow a new body underneath their existing body, because they can each segment can produce its own exoskeleton independently from those around it.
Yeah, there's no molting humans.
Right.
Unless you get sunburned, then there is. Because I definitely molted then.
All right, this is where we're talking about the specialization of the legs. Seeing some of the stuff that they switch into and the different functions that they are able to perform after they develop. I know it looks pretty bizarre. You got things with like whippy tendrils on them. You got things with spikes and claws. All sorts of functions available for these organisms. Some of these are used to help stir oxygen from the water into those feathery gills that we talked about earlier that existed underneath. They're called book gills. They're underneath the organism. So those are the predecessors to something called book lungs, which will be irreplaceable in the journey onto land for arachnids shortly after all this diversification.
They basically internalize the gills almost, right?
Mm-hmm.
Yeah.
Yep. Yeah. You can see that they're like an early Chelicera. They're all feathery on the outside, and as we move through time, they start to get up underneath, and you see them kind of internalize. It's really cool.
So we got normal lungs, and they got to read while they have their lungs, book lungs. They get to read.
Well, book lungs are the reason they're called book lungs is because they do have like pages in between them. They have to keep them moist, but they can absorb the oxygen and put it into the bloodstream. Or did I say bloodstream? I mean that again, speaking colloquially, it's really a hemolyth.
Hemolyth. Yeah.
Mm-hmm.
It's basically that their organs are bathed in a fluid that's like it's sloshing around a bit. I think they do have a heart, but it's not like closed. It's not a closed.
It's also
system. Yeah.
Its functions are multifaceted because hemolyth is also the liquid that they use to pump their hydrostatic skeleton to move. So different pressure levels on the hemolyth in different parts of the body is how they move their legs from one position to the other. That's fun stuff, huh? Did you guys know that?
In arachnids, well, in all or in most arthropods, is that okay? So I think.
Yeah, it's hydrostatic. Yeah, almost all arthropods. I don't want to say all because that's a kind of
I think in crustaceans there are muscles that attach to the outside exoskeleton, right? To as
yeah,
leverage.
well, within Chelicerata, that's they're it is hydrostatic because that's why they curl up when they die, right? Because there's no more pressure of the hemolyth. But there is a muscle structure. It's controlled by that ganglia. Each segment has 2 lobes on either side of it, right? And those lobes, the ganglia can send signals to expand or retract those little lobes. And when those lobes expand, it pushes the hemolyth that's around them, the same hemolyth that's carrying the oxygen, pushes it into pressurized chambers in the limb joints that cause them to move forward and backwards. It's very mechanical. It's very very weird. And there's this scientist who actually built a little robot thing with a needle that could tap into that hydraulic structure, and he could make robot spiders out of dead spiders. And yeah, it was pretty cool. It like injected this thing through the ganglia so he could control how it moves. I don't think he had it crawling or anything. He just had it grabbing stuff, but still. But yeah, with crustaceans, I'm not as confident at all about them being hydrostatic. I am confident about mandibulata being derived from hydrostatic organisms, though. Definitely, because they come from like organisms that were like velvet worms, right?
So what are we looking at? What are we looking at here?
So this is how different arthropod groups develop their limb structures. So you see what parts of the organism turn into what for each clade. Starting from the right, you got hexapods, which are insects, and crustaceans are next to them. And then we have myriapods. So those are your mandibulates right there. And you can see that they all have somewhat similar structures to how their limbs and things are laid out.
I do have a question.
Uh-huh.
Why is there a cross next to arthropoda? Aren't they still alive? Crosses usually mean they're dead.
Where are you seeing that? Are you talking about Artiopoda?
Oh,
Artiopoda is a little different.
Okay,
So
never mind then.
It's a yeah. But the 2 central ones are our chelicerates that we're looking at, right? So you can see what's developed into what. I like how there's a question mark by the Pycnogonida. It's like, what, we don't know what this is. But you see the yellow.
They are very weird. These sea spiders, yeah.
Mm-hmm.
And I think it's also still an open question whether they are close to chelicerates, or maybe they are even a group outside the mandibulate and chelicerate group.
Yeah, something basal, more basal, and I think Aaron actually found an article that said that they were maybe closer related to Artiopoda, which would be interesting. But yeah, so.
Oh, I actually have to go soon, so I thank you for having me.
No problem.
Will catch you later.
Yeah, thank you so much for being willing to pop in and hang out with us.
Yeah, you can always comment in the comment section later on stuff that you wanted to talk about.
All right. Bye, guys.
Bye-bye. So the red on here is the hypistoma-labrum complex. The yellow is antenna in the mandibulates, but in the chelicera, it's the chelicera. So that's what you're seeing in yellow there. Then you got the blue, and that is like an appendage. And then in the chelicerates, it's actually the pedipalps.
What? So are these like genes or are these like genes or what?
Yeah, it's showing which genes are controlling the growth of which limb type or which Hox genes are in control of the development here.
So do we know why these first three bit the dust?
Well, I mean, just didn't survive extinction events. I don't think there's anything inherent about their structure or anything. There was a reason that they didn't make it. Just happenstance of whatever niche they were filling and whatever impact the ecology that caused them to die out. So here we have some artistic interpretations.
Oh, so these aren't photographs?
No, they didn't actually go back and get real photos. But these are artistic interpretations of what early arachnids that first emerged from the sea may have looked like. Mm-hmm.
Would you have these? Would you have any? If you could, would you have these in your aquarium at your house?
I wish. They're pretty big. This is supposed to be Pulmonoscorpius, but I don't think it looks anything like it, according to the description of the image. Doesn't look anything like a scorpion. Let's say a terrestrial Eurypterid. Oh, never mind. It's labeled wrong. Okay, that's supposed to go down here on this next slide. Yeah, there's a giant land scorpion. All right, there's our Pulmonoscorpion, which is the one that I was talking about being the size of a squirrel. And it had long, narrow claws.
Is he hunting these guys down here?
I imagine that's the idea of the artist here. There's also been speculation that these were fishing claws, so that's an idea too. So it's hard to say for sure what they ate, but they had lungs. That's the important thing. And here's the actual fossil of the organism. I wish somebody could find me a real replica of this. I can't find it. It looks like I'm like bone clone and stuff. If there's any fossil that I could ever get, it'd be Pulmonoscorpion kirkinsonensis.
Now, is the whole thing the fossil, or just the rock, and the other parts are like artist stuff? All of this is the fossil that was found. Okay.
Everything you see, even the parts that are separated from the main fossil, those were just cut out separately from the main rock. Yeah, I would love a replica of this with like maybe a red velvet background to it. That would be amazing.
More family trees now.
Yeah, let's get into it. Let me see. We're looking at, which one are we on? Pantetra pulmonata. I'm able, if I can just type it into my document, I can pull it right up. Okay, let's take a Pantetra. Okay. So this is the group that contains whip scorpions, amblypygids, and arania, which are spiders. They tetra pulmonata, tetra, like tetrapod. It's talking about its legs. Pulmonata, pulmona is lungs. So legs and lungs are what we're looking at. Now there's two splits to tetra pulmonata, the schizotorsata and the sericodiastida. The arania is in the bottom plate that I'm not going to say again because that was a tongue twister enough, along with the extinct Uraranidia. And then the other group, you have Haptopoda, which is extinct, and then you have Pedipalpi, which has a lot of cool extant species, including the whip scorpions and tailless whip scorpions and all the amblypygids and Theliphonida. So that's these clades right here. As far as the Trigonotara beta, that's the one me and Ness were talking about earlier. That was very much like a giant mite that filled a niche like a spider, but it wasn't.
By the way, if anybody wants to see Bree's notes, they are in the description below.
Yeah.
If you ever want to check up and say, "Ooh, that's cool," or "That's wrong."
Yeah. Please tell me if I get anything wrong. Yeah, I'm not head in the clouds and not think I got all this right. I study this stuff for fun.
Yeah, unlike like some people, we can when we're wrong, we can admit it.
Yeah, I make probably lots of mistakes every day. I try to do better. All right. Probably got about 10 minutes more left that I can stick around with you. You want to see how many more slides we can get through? I think that's the same one we were just looking at. Ooh, Chimera arachne. This is the spider that still had a telson that they found in the amber. I believe it was in Myanmar. Let me see. Chimera arachne was found. Yep, Myanmar and Burmese amber or Myanmar amber. It is a I guess you could say it's closely related to spiders, but it sits outside of the all living spider clades. It doesn't belong to any of these extant groups, but it had a very, as you can see, a very unique armored appearance, like what we see in basal spiders, like the armored trapdoor spiders. Next slide. All right, so here we're looking at some venom discussion. So we have also the. Can you make that any bigger? I'm having a hard time seeing it.
Let me see if this will
slide pulled up. Oh yeah, I do. Here, I found my copy of it.
Any better?
Yeah. So the ones with the green are the ones that have that do produce venom. So you're talking about the pseudoscorpions, the scorpions, and the arania. Everything else on here, all other arachnids, are non-venomous. And as you can see how the clades are divided up, you have your tetrapulmonata, like we talked about a moment ago, with your arania, amblypygids, and Theliophanida. And then your scorpions are also included in there under arachnopulmonata. And of course, scorpions having their famously venomous sting, and pseudoscorpions having a venomous bite. And then we have the other branch of arachnida, which is Parasitiformes, Solifugida, Ricinulia. That's the clade that he was saying that they showed that horseshoe crabs were closely related to, interestingly enough. Opilions, and then a cariform. Our cariform is.
And the bottom ones are nuclear powered? Is that
yes. No, they're. Can't you see that they're toxic? That's not nuclear. Nuclear is like the triangles, right, with the circle in the middle. I hope I didn't get that wrong. That should be biohazard?
Either way, I'm staying away from it.
Yeah, I wouldn't go anywhere anything that says biohazard on it. All right, here's talking about some scorpion body parts. You have, starting from because it's hard to read, I see. Starting from the top left, you have carapace, and then the right is showing the chelicera. Also, you know what? I'll go down the left and then the right. So on the left is carapace, leg 1, leg 2, leg 3, leg 4. Then a metasoma segment 1, 2, 3, 4, 5, and the telson. And then going down the right side, we have the chelicera and the pedipalp, and then we show the divide between the prosoma and the opistoma. Then you got the different pterygoid segments, the metasomal pterygoid 1, metasomal pterygoid 2, metasomal pterygoid 3, metasomal pterygoid 4, metasomal pterygoid 5, mesosomal pterygoid 6. And then you got the mesosomal pterygoid 7, and also the legs 4, 5, 6, 7, 8. Yeah. And so forth. So those are the parts of our scorpion. All right. And here's another example of the development of different mouth parts among these different arthropods. And this one does include some tardigrades, so you can kind of see what's going on. Whereas in the velvet worms on the far left and the tardigrades, the front portion is the head. The second section on the velvet worms is actually the jaws. It's walking legs on tardigrades, and it's
nice.
slime glands is the next on the on the list for a velvet worm. Walking legs, more walking legs for tardigrades. Tardigrades love their walking legs. So moving into your arthropoda, we got chelicera, which is chelicerata, which is our topic of the day. Showing its head portion, then it's chelicera with its pinchy little parts. And then we got pedipalps behind that. Myriapoda, you got antenna, same thing with pancrustacea, which does include insects. And then behind the antenna in pancrustacea, you got antenna too. But in Myriapoda, you got intracalcary segments, which in centipedes, by the way, are forceptules that have venom glands on them called toxogontophorids, I believe is the proper terminology for them. Anyway, yep.
Next one.
All right, some embryology, some more embryology here. This is a development of the segments of the arachnid developing, and you can see that the legs, they start off with multiple legs, way more than the 8 that you expect from an arachnid before they fuse in development. So every arthropod is a centipede for a little bit. In a way, it's like, what do you call it? Recapitulation.
Evo Devo.
Mm-hmm. This is just another one like the one we looked at previously. Just it gives us more details. Now we are looking at mouth parts and antenna of different arthropods. I don't know if this is going to be relevant to what we're looking at here exactly, because cheliceras don't have antenna, so I'm not sure how why this slides even in here. Sorry, audience. Hate to disappoint.
The slide anything?
This slide is showing early development of arthropods and what we find in the Burgess Shale. Mm-hmm. More breakdown of the structure of mouth and body parts. These were really important to what we were trying to cover on the topic that we were talking about. But you want to just pop out and do some see if we got any questions from the audience before we wrap up?
Sure. Some of these last slides real fast.
Yeah.
And finally, this.
Yep, there's a nice cladogram for us. How to divide up your arachnids. Right. Spiders are ambush predators. Yes, that's true, Eddie.
Anybody have any questions?
Did you just kick me?
No.
Hi. There I am. Hey, does anybody have any questions? Let's see. Eddie's been dropping some questions or some commentary here. These creatures come in all different sizes, that's for sure. Yeah, chelicerates are a diverse group, and arachnids are a diverse group within that diverse group. Absolutely. And I find them all quite fascinating. He says spiders are ambush predators. That's absolutely true. Some spiders are pursuit predators, though, so we can't always shoehorn all of them into one category. The majority of spiders are ambush predators.
So how come none of these guys ever decided to become vegetarians?
There is a spider, jumping spider called something Kipling. It's named after Kipling from Jungle Book fame, Rudyard Kipling. But it's a jumping spider that lives off of seeds. So yeah, it's not. It's the only known spider that doesn't eat predatory or have a predatory diet. Now, harvestmen, opiliones, which Nestle had mentioned earlier, they also will eat like plant matter and stuff. So yeah.
Cool. Well, anything coming up on your channel that you want to advertise for?
Yeah. Tomorrow I got Creation Watch. We're returning to the Black Goo. That's tomorrow afternoon at 2:30. So don't miss it.
And for me, in about 2 weeks, I'll be having my season finale as we talk about the fall, the Roman Republic.
Woo! All right.
See how a republic became an empire, and sadly, it wasn't the same way it happened in Return of the Sith. No lightsaber battles on volcanoes, but it was still interesting.
Yeah.
We do have a question.
Oh, sorry, I didn't even have my screen up. Are there any non-venomous spiders? So all spiders are going to inject some sort of enzyme to break down their food because they don't have the mouth parts to eat solid food. So they have to liquefy whatever they're eating. That's generally the job of what we call venom. However, there are some species that are functionally non-venomous. Some like a hackle or weaver spiders, for instance, that have really good, strong webs. They don't have venom that's going to be too effective against anything. But they just need the digestive enzyme. But there is it's called the crebolate orb weaver or hackled orb weaver that is doesn't have the venom glands. It's lost them secondarily because it's just so good at catching its prey with silk. It doesn't need them. But yeah, it still does the digestive enzymes though, so there's still something happening there as far as having a toxic effect on the biology of its prey.
Cool. Well, I wish we had time to show the videos that you wanted to show, but maybe we can do that in a part 2 in the future.
Yeah, we can revisit that. I was just worried if there was going to be too much silence, but we talked through the whole thing, so that's fine.
Well, thanks everyone for coming. Thanks again for Bree for being on the channel again and showing his knowledge about his passion. Her passion.
All right, yeah. Thank you for having me on.
And as always, never stop learning. Enjoy the randomness. I'll see you next time. Bye.
Bye.